In this video, we will introduce real-time PCR, qPCR, a powerful technique for measuring DNA/RNA amounts. We'll cover principles, applications, components, and functions, as well as primer/probe design, RNA isolation, cDNA synthesis, quantification, analysis, and troubleshooting. Let's explore the world of qPCR. Real-time PCR, or qPCR, is a cutting-edge technique in molecular biology. It amplifies and quantifies DNA or RNA in a sample using fluorescent dyes or probes. This allows us to monitor the reaction in real time, revealing important reaction details. In qPCR, DNA or RNA is amplified and detected through a series of steps. DNA strands are separated, primers bind to target sequences, and DNA polymerase extends the primers, increasing the target sequence. Fluorescence measurements track this amplification process. In qPCR, there are different types of assays, each with its own benefits and uses. Cyber green-based qPCR and probe-based qPCR are the most commonly used assays. Cyber green dye binds to DNA and increases fluorescence, while probe-based qPCR uses specific probes that become fluorescent when bound to the target sequence. Assay selection is dependent on experiment needs, sensitivity, and specificity. In the section on qPCR instrumentation, we explore the essential components that enable accurate and efficient amplification and detection. These include the thermal cycler, detector, optics, and software. The thermal cycler controls temperature for denaturation, annealing, and extension. The detector measures fluorescence, while optics capture the signal. Software analyzes data, calculates CT, and generates amplification curves. All components are vital for reliable qPCR results. Designing primers and probes is crucial in qPCR, affecting the assay's accuracy. Primers must target unique sequences, avoiding non-target regions. Primers should be around 18 to 24 nucleotides long with a TM of 60 to 65 degrees Celsius. Probes, like TaqMan probes, emit fluorescence when they bind to the target sequence. Thoughtful design guarantees precise quantification. Before performing qPCR, it is essential to isolate high-quality RNA from the biological sample of interest. RNA isolation methods vary depending on the sample type, with common techniques including organic extraction, column-based purification, and magnetic bead-based methods. Once RNA is isolated, it is converted into complementary DNA, cDNA, using reverse transcription. This step involves the use of reverse transcriptase and primers to synthesize cDNA from the RNA template. The resulting cDNA serves as the starting material for qPCR amplification. Absolute quantification in qPCR uses a standard curve to determine the exact amount of target DNA or RNA molecules. Standard curve is generated by amplifying known concentrations of a reference template. CT values of standards are plotted against known concentrations to interpolate target molecule amount. Relative quantification in qPCR compares gene expression levels between samples. It uses reference genes to normalize data and calculate fold change in gene expression. This method provides insights into expression patterns and is important in gene expression studies. Several factors can influence the efficiency and accuracy of qPCR. These include primer and probe design, primer concentrations, annealing temperature optimization, template quality and quantity, choice of master mix, and cycling conditions. Careful consideration of these factors and appropriate optimization of the qPCR conditions are crucial to ensure reliable results. Small variations in experimental parameters can significantly affect the efficiency and specificity of the reaction, emphasizing the importance of meticulous experimental design and optimization. qPCR data analysis involves extracting meaningful information from the amplification curves and CT values obtained during the experiment. Several algorithms and software programs are available to analyze the data and calculate the relative gene expression levels or absolute quantities. These include the 2 to the power of minus delta delta CT method, comparative CT method, efficiency-corrected methods, and advanced statistical analysis tools. Careful analysis and interpretation of the data are essential for drawing valid conclusions and making accurate inferences about the gene expression or target molecule quantity. qPCR has found extensive applications in various areas of molecular biology research. Some of the most common applications include gene expression analysis, pathogen detection, genetic testing, molecular diagnostics, microbial quantification, and environmental monitoring. Its high sensitivity, specificity, and wide dynamic range make qPCR a versatile tool for studying gene regulation, disease mechanisms, biomarker discovery, and personalized medicine. Its widespread use across different research fields demonstrates the broad impact and significance of qPCR in advancing scientific knowledge. Despite its many advantages, qPCR can encounter technical challenges that may affect the reliability and accuracy of the results. Common issues include the presence of inhibitors, suboptimal primer and probe design, contamination, and incorrect cycling conditions. Troubleshooting these problems requires a systematic approach, involving optimization of reaction conditions, assessment of reagents and equipment, and careful experimental design. By understanding the potential pitfalls and employing appropriate troubleshooting strategies, one can overcome these challenges and obtain robust and reproducible qPCR results. In conclusion, real-time PCR, qPCR, is a powerful technique in molecular biology research that allows accurate measurement of DNA or RNA in a sample. This video covered all the necessary details and concepts for medical students and researchers to use qPCR effectively in their work. By understanding the basics, principles, and troubleshooting strategies, they can utilize qPCR to enhance their understanding of molecular biology and contribute to scientific advancements. As technology advances, qPCR will remain a vital tool in diagnostics, personalized medicine, and biomedical research.